know it's we take advantage of our summers to get all those things done that we can't get done during the school year, and we certainly do appreciate you taking the time to come and listen to our seminar speaker today, Dr. Gavin Conant, who is being considered for a faculty position in biological sciences. Presently, Gavin is an associate professor. He's in the Division of Animal Sciences and affiliated with the Bioinformatics Institute at the University of Missouri in Columbia. His PhD was obtained in biology from the University of New Mexico. He did a couple of postdocs. He started a postdoc in Leipzig University in Germany. And while being involved in that postdoc, he, what I interpreted, at least in our conversation, was like his dream postdoctoral position became available in Trinity College in Dublin, working under Ken Wolfe. He applied and was offered and took that position, so he left and finished up his postdoctoral studies in Dublin. Gavin's interests are in computational evolutionary biology. He's interested in understanding how or what drives the processes that are responsible for evolution. He studies these at the genome level. If you were to look at his CV, you would see that he's done a lot of work in agricultural related commodity organisms like broccoli and cows and things like that. And I think that reflects where he comes from. It's the division he was in to a large degree, where the money was coming from. But he certainly did convince me that he's a basic scientist. He's interested in basic processes. He studied these processes in a variety of species, since the commonality among all of us is that we have DNA, and that's what he's interested in. So if you do get a chance to look at his CV, you have, don't be put off by the ag slant. He definitely belongs in biological sciences and not in a cows department, at least as far as I'm concerned. A reminder to any of you that would like to spend a little time with Gavin, he will be in the conference room over at Thomas from 9 to 10 tomorrow. Please, I encourage you to stop in, chat a little bit, hear more about his research, hear more about him. We will have coffee for you. So the title of the seminar today is Life Will Find a Way, Network Insights into Ecological and Evolutionary Complexity.
Great, thank you very much. And yes, thank you all for taking time out of, I'm sure, many things you had to do over the summer to come and listen to this. Hopefully it's worth your while. But Life Will Find a Way is a joke from Jurassic Park. It's not a very good joke, but it was what I could come up with. I'm sure if you're wondering about that. The second part is actually true. So rather than trying to rush for this at the end and doing it badly, let's just do it right at the beginning. Obviously, this is work that I did with a lot of people who had a lot of help. Backwards, I'm going to talk about this part of the work first and this part of the work second. So the first half of the talk is some yeast evolutionary genomics, studying the yeast polyploidy event. The underlying data for that is very generously provided by my postdoc advisor, Ken, and two of the people in his either former lab members, John, or a current lab member, Kevin, who's actually his lab manager. So none of that work would have been possible without their sort of generous giving away of the base data. And then some friends and colleagues at Missouri, Andy and Dimitri, and Jim and Chris, helped me sort of frame the questions for that data, for that part of the talk. So I'm very appreciative of that. The second part of the talk is the metagenomics half. That's part of a big project that's actually being, the PI that ran is Christy Kamek, who was at the University of Wyoming, is now, I believe, at the University of South Dakota. This is all very new information. She moved. Her graduate student, Belinda, actually did the actual touching of animals, because I don't touch animals. Bad things happen when I do that. So she actually did the sampling, the DNA extraction, all of that. My master's student, Sarah, with the assistance of Tasia, who was another PhD student in our department, actually did most of the computational analysis for that part of the project. And then Bill is my collaborator at Missouri, helped me with that project. And then Yue is another student in my lab, and Juan, they helped with some pieces of that analysis that I'll mention as I go along. And then my wife, Michaela, helped me and actually Juan port part of the code that we did the metagenomic analysis onto a GPU. So that was actually pretty cool. So if you're the ways we could use new and emerging computer hardware to do biology. You could definitely be interested in her. The yeast work is sort of funded off of the bits and cheese pairings I can find in my department. We have an NSF grant to do some of the computing stuff, and then another NSF grant to essentially repeat the yeast work in broccoli. I'll mention that at the end of the day. And then the USDA actually funded the metagenome. Okay, so that actually kind of leads into first the outline of the talk. I want to tell you a little bit generally about why the lab seems so schizophrenic. So that's the sort of our lab in background. And then just two samples of what we do. A yeast comparative genomics analysis, a sort of deep evolutionary genomics, looking at polyploidy, the evolution through functions, modeling, a few interesting things like that. And then a metagenomics component where we're trying to understand essentially how you build an ecosystem inside an animal and what that ecosystem can do. Can you understand that? And since I'm not an ecologist, part of this is begging for help from an ecologist saying, how do we understand these data? Yes, I can analyze all this genomic data, but what do you do with it once you sort of put it together into some kind of framework? So, a couple of years ago, I decided to be pretentious and retitle our lab the Evolutionary Systems Lab. I'm still a little unsure whether that was a good move or a bad move. The reason I did that is I wanted to get the idea that we work not so much on necessarily always on systems biology, but nonetheless with the idea that we're always looking at systems that have an evolved history. And so even when we're talking about metagenomics, we're talking about A, a group of organisms that have their own individual evolutionary histories, and then B, a host organism that's evolved to sort of host those things, and how is that a system? Is it a self-assembling system? Are there rules, or can we understand how it gets put together? So we have in the lab a lot of money that are sort of pieces of other people's grant, and that's actually one thing I'm very conscious of. That's one thing I'd like to change, is have a little bit more, a few more projects that are directly funded out of our lab. But the big projects we have in the lab, the metagenomics I already mentioned. Chris Piers is the PI of a plant genome grant from NSF that's looking at the genome triplication that's common to the broccoli, cauliflower, cabbage group, but also the kale, Chinese cabbage group. They share a genome triplication, along with a number of other wild species that certainly I've never heard of. And so the question that I'm going to do a lot of yeast comparative genomics, all the toolkit that we built for the yeast comparative genomics, polyploidy analysis in yeast, we're just going to port those straight over and do the same thing with these broccolis. One of the questions being, for instance, is the fact that we could domesticate broccoli and cauliflower, and the fact that broccoli and cauliflower and Brussels sprouts are all the same species, which is an amazing thought if you think about it. Does that have anything to do with the fact there is this genome triplication that gives it the plasticity to be domesticatable and to also have this sort of phenotypic variation? Michaela and I have a grant together from the computer side of NSF to essentially take new emerging hardware platforms from the computing point of view and try and apply them to biological problems, so GPUs, but also some other things that I'd never heard of called the microautomata processor, which is, I'll tell you about that if you're interested. We also have some EU money, which I don't really want to talk about because it's very confusing, to do essentially some comparative genomics in the mammals as opposed to comparative genomics in the broccoli or the yeasts. And then the grant that I'm the PI on, which is ending this summer, is a 3-year NSF RU program. The way that works is we bring 12 undergraduates from more or less across the country to NU. We pair them up into 6 teams of 2, and we put them in labs to do research. And what we try to do is we pair off a computer scientist with a biologist, and we put them in a lab that, say, has some data that needs to be analyzed. That lab may not have a lot of computational experience. That CS student may not know anything about biology, but he talks to his friend who's a biologist, and they write a Perl script that does something useful for that lab. We've done that for 2 years now. This is the 3rd year. It's incredibly rewarding to actually do that, to interact with those undergrads. It's a little bit of a nightmare to actually get them to apply and run through all the applications and that kind of thing. And so we're not sure whether we're going to do this again, but it was a great experience. And so we're trying to think about ways we could do something like this that doesn't involve quite so much bookkeeping. Okay. So what we're actually doing is what I already mentioned, a sort of one vignette on the genome duplication in yeast and a second one on metagenomics. So my former postdoc advisor, Ken, shortly after the yeast genome came out in '96, published a paper that showed that there had been a genome duplication in Baker's yeast and several of its close relatives. A really nice paper came out in PLOS Biology last year that actually showed that what really happened, because it had always been thought, but it had been very, very difficult to prove because this event happened about 100 million years ago. But they were able to show that the event was actually the merging of the genomes of two related yeast species, but nonetheless not the same species. So if you've heard the lingo, that's what's called an allopolyploidy as opposed to a quadruploidy, where you bring together two identical genomes. It had always been hard to tell the difference between those two because the event was so long ago, it was hard to see whether there was subtle differences between the two genomes that came together. Irregardless of that difference, what effectively happened is you went in one generation from a genome of 5,000 genes to a genome of 10,000 genes. And so you have effectively two copies of every gene. Because it's an allopolyploidy, those two copies are probably not absolutely identical, but they're quite similar. One of the things that I didn't understand about polyploidy until I started to read about it is this situation of having a double genome, of having a second copy of every gene, is actually quite evolutionarily unstable. It decays quite quickly. So most of those 10,000 duplicates no longer exist in any of these yeast genomes. So if you're following back the envelope numbers, the current Baker's yeast genome has 5,500 genes. So it essentially has an extra 500 duplicate genes on top of a sort of quote-unquote normal yeast genome that are attributable to this event. So for the rest of this half of the talk, all I'm going to be talking about is trying to understand why 4,500 duplicate genes disappeared and why 500 duplicate genes in the yeast genome are still kicking around. What are the forces that drive that process? And I'm going to argue there's a number of them, but we'll sort of get there as we come. Okay. So here are 12 species that all share the genome duplication. So you can see as I've got a mirrored phylogeny, the same 12 species down here as up there, and you can sort of walk through the genome and start to talk about cases where one of the two copies of the gene, so from parent one of the allopolyploidy, survive in all the extant lineages, and so here is the second parent of the allopolyploid. So you can think of this as being species A that came together to form the allopolyploid. This is species B. What you can see is for this column of genes, everybody kept the copy of parent A, and everybody lost the parent of copy B, the copy from parent B, excuse me. So to put the big word on that, we would call these orthologous genes. I'm going to explain where this figure came from in just a moment, because it's actually not trivial to get to the point where you can claim that these are all orthologs. And I'll talk about that in a minute. Another thing that can happen is you can have alternative losses. So these 9 species decided to keep the parent B copy, but these 3 species kept the parent A copy. So we call that reciprocal gene loss. Somewhat rare is the situation where everybody keeps both copies. And I'm going to talk in a minute about why this might happen, what can drive the retention of the duplicate gene across one or more genomes. Okay. So the trick to all of this is we just get these genomes as a list of genes. We can identify essentially these columns by homology, by blast search. But what becomes tricky is to decide, is this gene here in Saccharomyces cerevisiae the ortholog of this gene, or is it potentially the ortholog of the other copy in the other genome? And sort of essentially to phase all the genes to decide this is an orthologous block of genes and this is an orthologous block of genes. It's a little bit tricky, and so we developed a software tool called POINT, which stands for bad name, so it's essentially Polyploidy Orthology Inference Tool, just so we have a name to put to it. What that does is it not only does this phasing, it says this group of genes is orthologous and this group of genes is orthologous, but it also gives you a probability for that orthology assignment. So those numbers you see across the top are its estimate of how certain I am that this gene and this gene are, that number says I am 99.1% confident that these 2 genes are orthologs and these 2 genes are orthologs, rather than this PAP2 gene being the ortholog of this gene right here. So we built that tool when I was a postdoc. We sort of expanded it and improved it later on. And so everything I'm going to talk about from here on out is based on using this framework to find the orthologs. And then I'm going to argue with you that I can use, the fact that I can find all those orthologs allows me to do a lot of interesting things analyzing the genome duplication that I wouldn't be able to do if I hadn't put it into this framework. All right, so but the big question is really, why do we, you could look at it two ways. Your temptation is to look at it as in the direction of why are we losing genes? What genes are going away? The danger with that framework is that a lot of genes are going away effectively by drift. And the idea being the following. We're simply throwing mutational bullets at the genome and disabling genes. In the case of a genome duplication, if you've got 2 copies of every gene, don't worry about haploid versus diploid for the moment. If you've got 2 copies of every gene, you can effectively lose one, arguably with no selective cost. So if we frame our question of why are we losing genes, we're being a little bit confused because we're losing a lot of genes by mutation. We may not be losing all the genes by mutation. There may actually be quote-unquote selective losses, and I may come back to that in a minute. But a better way to frame the question might be to say, not why are we losing genes, but why are we not losing certain genes? So we're firing mutational bullets in the genome all the time. We're disabling a bunch of genes. Most of the time that's tolerated, and the gene just disappears over evolutionary time. But in some cases, it's not allowed to use the gene. Selection disfavors individuals that have a mutation in one of the 2 copies. There's a whole area of evolutionary biology that focuses on this question of why do we have duplicate genes in the genome? What preserves them? And there's several interesting ideas. The oldest is what's called neofunctionalization. So that's saying we had a duplication, one of the 2 copies of the gene took on a new function. They are now both selectively necessary for the organism, so you can't lose either copy. A canonical example of that might be like the hemoglobin gene, where one functions in, actually that's probably a bad example. When you start to look more closely, you find that these things get much more subtle than I've made them out here. But you can imagine, say, for instance, an enzyme ancestrally had one substrate, you have a gene duplication, one of the 2 copies becomes specialized in a new substrate. You now have 2 genes, one that does the old substrate, one that does a new substrate. Neither of them can be lost because they're both necessary. That would be a neofunctionalization. Another cool idea that Alan Force and Mike Lynch and others had a few years ago was the idea that you might have had an ancestrally multifunctional gene, so say a gene that functions in 2 tissues. You duplicate that gene, and then you subspecialize each copy, one to one tissue and one to the other tissue. There's other ways to do subfunctionalization, but that's a nice mental model. Now both copies are necessary because if you lose either copy, you lose expression of that gene in one of the 2 tissues. So that would be another way in which 2 duplicate genes could become both selectively necessary, and you wouldn't lose either copy. I rather unfortunately contributed to some nasty terminology problems by naively using the word dosage in a paper when I shouldn't have done. So you can talk about how much of a gene product is being made, and in a sort of simple null model, when you make a gene copy, when you make a gene duplication, you're potentially increasing the expression of that gene because you're expressing both copies. That could not be true for a number of reasons, but just bear with me. You could imagine that there simply be selection on that it's simply beneficial to overexpress a gene, and one of the ways you can overexpress a gene is just to have 2 copies of it. There are other ways, but if this is the first mutation that happens, that might be the way the organism has chosen to overexpress that gene. There are known examples of that, things like resistance genes in plants, amylase copies in humans and dogs. So this is what I call selection on absolute gene dosage. All I mean by that is just saying this gene in isolation benefits from an increase in expression. And again, you could achieve that increase in expression in different ways, but I'm just going to claim one of the ways you could achieve it is by gene duplication. Again, that could preserve a different gene. An idea that I had to have explained to me about 3 times before it finally penetrated me and my brain, and I actually said, oh, that's a great idea, was there's also a second kind of gene dosage, and that's what I call relative gene dosage. And what that means is if you've got a group of genes that work together, they may make a protein complex, they may be part of a pathway or a cascade or something like that, and you start to alter their relative abundances, that can cause problems. So you can imagine if you want to build a dimer, 1 copy of A, 1 copy of B, and you suddenly dramatically increase the expression of A relative to B, at a minimum, you've got a bunch of A monomers floating around that aren't doing anything. Arguably, depending on how the complex assembles itself, those A monomers might actually get in the way, and you might end up with not enough AB dimers. There's a whole series of ways that this can happen, and there's actually tons of great review papers if you're curious. But this idea that you may need to keep things in balance. Another example might be like a transcription factor that's a repressor. You suddenly duplicate the transcription factor, and you over-repress a bunch of its target genes because it's being expressed too heavily. So that's another case where the expression of one gene relative to the expression of a bunch of other genes needs to be maintained in balance. Okay. One other idea that I just recently come across in the literature from Razier and O'Shea is you could actually maintain a gene duplication to reduce the expression noise of a gene. So if you can imagine a gene is being sort of stochastically turned on and off, transcribed on and off. And so what that means is you get fluctuations in the mRNA levels. That's sort of intrinsic gene expression noise. You can't get rid of that no matter what you do. But if you duplicate the gene, you're now expressing the gene from two places, and actually the variance in the mRNA levels will decrease. And if that's somehow selectively beneficial, you could actually preserve a duplication for this. I don't think there are any known examples of this. This is just a hypothesis, and one of the things I'm interested in the future is trying to test that hypothesis. Okay. I suspect that all of these things are happening in the yeast genome after the polyploidy. That genes are being preserved for all 5 of these reasons, but I'm really going to focus very heavily on this gene dosage idea because I think it's one of the less well-studied and more interesting reasons we can preserve duplicate genes. Okay. So the name this goes by is the dosage balance hypothesis, and I just actually described this to you. We've got genes that work together. That doesn't have to be they physically interact. It could be transcription factors and their targets. It could even be a metabolic pathway if that metabolic pathway is sort of odd kinetics. But all it's saying is that we can't start to increase or decrease the level of one gene in that system without affecting the whole system and possibly in a bad way. So this is an idea that's due to a lot of really clever people. Jim Birchler at MU, Mike Freeling, Lawrence Hurst, and others have all written really nice reviews on this. And so this is an idea that's sort of been kicking around in various forms of literature for a really long time. But in the genomic world, probably since about the early 2000s, people have been talking about this as an explanation for why polyploid genomes in particular look the way they do. So we said, if this is one of or a major force that's preserving the duplicate genes in the yeast genome, then when we go look at those 500 surviving duplicates or the other surviving duplicates in other lineages, we would predict that the ones that survive in duplicate are the ones that interact with each other, or at least interact with a lot of other things. So a testable hypothesis from this hypothesis, a little bit nested there, is that there should be a trend in which genes survive in duplicate based on their degree of interactivity with the rest of the genome. And what you mean by interactivity could vary, and we'll come to that in just a minute. So I'm actually not really going to talk about this. I'm just going to say this was an analysis we did looking at the yeast protein interaction network, trying to understand how interactive it actually was. And I think the take-home message was there's a lot of subtle things that happen because proteins bind to each other that contribute not only to sort of how the cell is spatially organized, but contribute again to this question of balance and saying that it's not that genes function in isolation. They function in pathways, they function in complexes. And that probably matters a lot for understanding the genome duplication. Okay, so I said that we use that point tool to infer the orthologs. But the other thing that it allows us to do, which hadn't really been possible in other models, sorry, other groups that have polyploidy, is to look not just at the extant genome. So we can look at any 2 extant genomes that have a polyploid event and say, oh, there's these 20,000 surviving duplicates in Arabidopsis, or 5,000, or whatever the number is. There's these 500 surviving duplicates in Baker's yeast, ... But because we have 12 genomes that all descend from the same polyploid event, we can put them in a phylogeny, we can put them through a model of evolution, and we can talk not about just what survives to the present, we can go back to any arbitrary point in the past since the genome duplication, and essentially model the genome that existed at that point in the past. And so that says, we can say, when did a particular gene get lost? Not that it got lost sometime between the polyploidy and now, but actually put a particular phylogenetic branch and say the loss was on that branch. And so that allows us to actually look for evidence of selection and dosage, but not really in the extant genomes, but in the history of the resolution of the genome duplication. So we do that, and essentially, we infer a phylogeny, except this is not a gene phylogeny. This is not a phylogeny from gene sequence data. This is a phylogeny from duplicate genes. And so what these branch lengths are proportional to is not some sort of measure of DNA or amino acid change, but if you were a duplicate gene here, how likely is it that you were still duplicated here? So here at the very root of the tree is the genome duplication. This long branch is saying in this genome, almost all the duplicates have been lost. Because we started out way over here with a few duplicates, and then the longer this branch went on, the more of those duplicates returned to single copy. And so what these numbers above each branch indicate is the number of duplicate genes that will return to single copy along that branch. So what this is saying is the duplication happened here. 500 of the 5,000 genes, or the 10,000 genes, will return to single copy here, and then there was a speciation between this clade and the other clade. So what that's saying is there's this speciation right here was very, very soon after the genome duplication, to the extent that these genomes are almost completely different in what survives in duplicate and what is single copy than these 9 genomes. So what we can see is most of the losses were happening in these 2 branches. So by the time you get to this branch plus this branch plus this branch, we only have about 2,000 surviving duplicate genes here, down to even fewer than that here. And so when we get out to the extant S. cerevisiae genome, these numbers I should say, not all the 5,000 genes were in our model for various uninteresting reasons, so these numbers don't add up to 5,000, and I apologize for that. So by the time we get out to cerevisiae, very few genes were lost between after this last speciation between these 2 close related yeast species. Okay. So I think this is cool, but that's just because I wrote it, so probably no one else does. But the idea that we can actually figure out not merely that this gene is single copy in this species and a single copy in this species and single copy in this species, but start to figure out when a gene loss event occurred on this phylogeny turns out to be pretty useful. And the way we do that, just so in case you're curious, is we have a model. And this is nothing more complicated than the models you're used to seeing when you do the HKY model in PhyML and the GTR model in PhyML. If you've done phylogenetics, these are the same models you use, except that instead of the states being A, T, G, and C, the states have to do with whether a duplicated gene is duplicated, it's returned to single copy 2, it's returned to single copy 1, or it's become a fixed duplicate. Don't worry about these 2 other states for the moment. So the way this model works is all 5,000 loci start in state D. They're all duplicated by the polyploidy event, by the genome duplication. Some of them lose copy 2 to become fixed to copy 1. Some of them lose copy 1 to become fixed to copy 2. A fraction of them become fixed duplicates. You can think of that as like neo-functionalization or sub-functionalization. You fit all these parameters in your model by maximum likelihood. And when you do that, you can then, for every branch of the tree, infer which losses occurred. So you can essentially say, what is the probability that a particular gene was duplicated here, but it returned to, say, copy 1 by here? You can sum up all those numbers, and those give you these loss numbers that we see here. But you can do something a little more interesting than that. And that is you can say, okay, what does that say about, say, the structure of biochemical networks at different points in the tree? So what I've done here is I've made a diagram of the modern Saccharomyces cerevisiae protein interaction network. So each one of these little dots represents a S. cerevisiae protein, and although you can't see this, and I apologize, there's a bunch of little gray lines that join pairs of dots. That's to say those 2 proteins interact with each other. Everything that's colored purple is duplicated, and what we can see is, right after the genome duplication, lots of things are duplicated, as expected. So all of the proteins are duplicated right after the genome duplication. But as we walk through the evolutionary history, we start to find these blue dots, and those are things that have returned to single copy. Okay, nothing very deep there so far, but if you squint and you look at these 3 diagrams, you can see that effectively the losses start at the periphery of the network, where there aren't very many interactions, and move towards the center. And so what that's saying, and we can actually show this statistically through other means, is that the earliest losses right after the genome duplication were of lowly interacting proteins, proteins that interacted with only 1 or 2 other proteins. And it was only later in the resolution of the genome duplication that we started to lose these core or highly interacting proteins in the center of the network. And of course, that was exactly the hypothesis we started with, which was it should be easy to lose duplicates that don't interact with any other proteins, because they're not constrained by dosage balance. that these proteins in the middle that interact with a lot of other things, we start to take them out of duplicate, that throws the balance of the network off. And so we were able to show that exactly the prediction of the dosage balance hypothesis is what we see in the real data when we trace not just the extant genomes, but we trace the history of the network through the genome duplication. Okay. So that was nice. We sort of validated the dosage balance hypothesis. Not that there was much doubt about it, but it had not been validated in this sort of time-dependent way before. But we actually found something even more cool, and this is sort of the new part. We went and we looked at pairs of proteins that interact with each other. So A physically binds to B. And we said, how likely is it that both A and B return to single copy on the same branch of the tree? Because that's the real test, right? It's not just are they highly interacting, but we're curious about pairs of proteins that are sort of working with each other. Do they need to be in the same copy number? They can both be duplicated, they can both be single copy, but if one is duplicated and one is single copy, that's a problem. That's what the dosage balance hypothesis predicts. And we went to see, do we actually see that? And the answer is yes. And so on every one of these branches in this tree that's colored red, what we find is an excess of losses of pairs of interacting proteins. So pairs of proteins that physically bind each other in the modern S. cerevisiae were both duplicated here, but had both returned a single copy here. And on this branch, there are too many of those losses to be explained by chance. And so at an even finer resolution than just the previous analysis, that suggests to us that this idea of dosage balance is important. That you can't have one copy. Now, obviously, in some cases you can, but there are at least some circumstances where having one copy of dimer, for instance, in double copy and one copy in single copy is not evolutionarily stable. Having them both duplicate is okay. Having them both single copy is okay. But once one has gone to single copy, there becomes probably selection for the second one to return to single copy. So that had not been seen before, so we thought that was exciting. That is not the only predictor of what remains in duplicate. I'm not going to bore you with too many details of this, but another thing that predicts whether a gene survives a duplicate or not is whether it is a gene that's highly regulated by a bunch of transcription factors. And so, in a somewhat complicated way, what this figure is trying to show you is, particularly in the extant genomes, the duplicate genes are regulated by more transcription factors than are these blue single copy genes. So another constraint on whether you can return to single copy after a polyploidy event is how complex your regulation is. If you're a highly regulated gene, it's hard for you to return to single copy again because that probably induces a bunch of imbalance in that regulatory network. You can imagine if you're a duplicate gene and you're being regulated by a bunch of transcription factors, and then one of your two copies goes away, then all of those transcription factors are trying to act on the other copy, and that could potentially be bad. So that was one predictor. And then the other thing that we found that was interesting is we previously sort of looked at metabolism in Saccharomyces cerevisiae, baker's yeast, because it's kind of a weird yeast. The reason we love baker's yeast is because it will make beer, it will make alcohol, and particularly it will make alcohol in the presence of oxygen, which most species would prefer not to do. And the reason they prefer not to do that is energetically it seems more efficient to run your glucose all the way through the TCA cycle and actually get all the ATP you can get by turning that glucose into CO2 water vapor. Baker's yeast doesn't do that. It turns its glucose into ethanol, even in the presence of oxygen. And it's probably the case that the genome duplication contributed to that phenotype. This is a very messy figure. All this figure is really trying to show you is that that sort of metabolic change that was produced by the genome duplication is probably a latecomer. So essentially, early in the history of the genome duplication, so this is the point of the genome duplication, this is early in the history, there wasn't much selection on metabolism to preserve duplicate genes. But recently, and particularly in this clade up here, selection on metabolism became important. So the genome duplication happened back here. There's a bunch of things that happened probably to the dosage balance and that kind of thing, sort of in the intermediate history of the tree in this area. And then very recently, probably, in this clade and this clade in particular, the remaining duplicate genes that survived from the genome duplication probably got repurposed to do some interesting things relative to glucose metabolism. But what's interesting is you can imagine there's some other species down here in this clade that didn't see that same change. They had the same genome duplication, but they didn't decide to use the genome duplication to change their metabolism in the same way that these yeasts did out here. So, agree with me or not, the claim we made in this paper, therefore, was that there were stages to the resolution of the genome duplication. There was a very early stage where just a few genes were lost, but this is pretty interesting because if you look across a wide variety of plant polyploids, DNA repair enzymes and mitochondrially targeted proteins are rarely found in duplicate in any plant genome, even though that plant genome may have 4 or 5 polyploids in its history. And so we saw that too. And so there seems to be something about genes with these functions that don't tolerate being a duplicate in a polyploid. So one of the 2 copies gets kicked out almost immediately. And I tend to think of that as basically being there's a cassette of mitochondrially targeted proteins. And you can pick parent A or you can pick parent B, but you can't mix them. And so what happens is soon after the polyploid, one of the 2 cassettes of mitochondrially targeted genes or DNA repair enzyme gets returned to single copy, gets eliminated, because the 2 copies don't function together. So that's, I think, a pretty interesting area of research where this is still really needs to be followed up. They're early losses, they're probably selected losses in the sense that it's not just that we lose this through genetic drift, but there may actually be selection favoring the loss of, say, the DNA repair enzymes from one of the 2 parents. We don't know how to prove that yet, so we're pretty interested in proving that, but I don't know how. So that's sort of stage 1 of the genome duplication resolution. Stage 2 is all that stuff I talked about, losing pairs of interacting proteins. That's the dosage balance stage. So the intermediate period after the genome duplication, if you want to, you can put numbers on it, say 5 to 25 million years. And I'm just making those numbers up, but approximately that. That's when dosage balance is really acting. And then after everything has sort of shaken itself out and the genome sort of re-diploidized itself, you have the late phase of the genome duplication. And that's potentially when you start to use the surviving duplicates to do innovation. So you have this thing, it was polyploid, it threw away most of the extra genes, it's just got a few % of retained duplicate genes, but those few % are in targeted pathways, and maybe you can actually use those retained duplicates to evolve something new. You can evolve glucose fermentation in yeast, you can evolve broccoli versus cauliflower, that kind of thing. So this is maybe the most evolutionarily interesting thing, but it only happens after the sort of initial phases get taken care of. So we call that 3-phase resolution of polyploid, whether it's really 3 phases or 4 or 2. That's probably a bit of a philosophical question. Okay, so shifting gears a bit. Talk about metagenomics. So why are gut metagenomes, and particularly gut metagenomes of ruminants, interesting? Well, A, because I'm in an animal science department, and that's the kind of thing we care about. But even if you're not, one of the major contributors to global warming is the methane produced by cow farming. And the things that produce the methane are microbes. And the farmers don't like this, even if they don't believe in global warming, they don't like this, because it actually wastes their food. Essentially, all that methane that the cow is producing is food that the cow is eating but is not using for growth. So this is one of those times where you can actually get a little bit of synergy between people who aren't really keen on global warming but still want to reduce methane emissions for their own reasons. So you can care about it for that reason. In principle, this should be a great biofuel. You can figure out how the cow is turning grass into short-chain fatty acids. You could potentially do the same thing in the reactor and produce fuel for your cars or your planes or whatever. It may have implications if you want to understand the human gut microbiome, although obviously that's not quite the same metabolic set of reactions, but still it may have, there's still the tools and analysis framework you build may actually be useful there as well. So I think it's an interesting problem. I just think it's also an interesting little contained ecosystem that you can study. And so the experimental design was as follows. And of course I say we because I didn't have a thing to do with this. I don't know how to touch animals. But they took 16 sheep, 8 of which were fed sort of a conventional sheep diet, a grass-like diet, and 8 of which were fed grains, in this case maize. And they sampled the rumen fluid from these 16 animals. They extracted DNA from that. They sent it off to the DNA sequencing court at the University of Missouri. We ran it through our Illumina machine, and we got back, I don't even know how many databases of sequence, but a lot of databases of sequence. So what do you do with that? Well, there's a lot of different things you can do. we did two specific things. We actually looked at what 16S genes were there, and that's sort of a taxonomy thing, and I'll show you that in a minute. But then we also looked at what enzymes were there, and that's what I'm going to spend most of my time talking about. One more thing I should say is the animals we chose to sequence were ones that gained a lot of weight for the amount of food they ate, so 4 that gained a lot of weight, 4 that gained a lot of weight, and 4 that gained very little weight for the amount of food they ate, 4 here and 4 here. So essentially, animals that were efficient in turning food into body mass, and animals that were inefficient in turning food into body mass. And that'll come back up in a minute. So one thing you can do with these kind of data is you can just see sort of what bacterial species are present. And so these were the 8 animals that were fed grass, and these were the 8 animals that were fed corn. And indeed, there are different species of bacteria present on average in this group versus this group. But we were sort of thinking maybe this isn't the right way to look at these data. We're sort of curious about not so much what taxa are present, but what biochemical reactions are present. And we said, what if we put this in a metabolic network framework? All a metabolic network is either a list of reactions and the compounds that join them, or a list of compounds and the reactions that join them. So we're going to focus on this. And so each of the little circles in our networks that you're going to see in a few minutes is going to be a reaction. You can assume that that reaction is catalyzed by an enzyme, one or more enzymes. And we join up 2 enzymes, 2 reactions, if they share a metabolite. So this is an approximation. This is an, what's the best word? A simplification of the real biology. We're not talking about flux, we're not talking about flow, we're not talking about directionality of pathways. We're just saying these 2 reactions are sort of related to each other because they have a common metabolite. That's an approximation, but maybe it's a useful one. So why did we decide we want to do that? Well, we had 2 cows that we fed sort of the same diet. And we looked at the correlation animal to animal of what species of bacteria were present. And there's a correlation, but I wouldn't say it's hugely strong. On the other hand, we took the same 2 animals, we looked at the correlation to what enzymes were present, and I would argue that it's quite a bit stronger. So what this is saying is there may be a bunch of different species of bacteria that are doing approximately the same metabolic thing. And so when you look at the taxa that are present, you say, oh, these 2 animals are different. But when you look at the enzymes that are present, you say, no, these 2 animals are doing approximately the same thing. Another way to think about this is you've got your forest, you've got a whole bunch of different tree species, but maybe some tree species fill the equivalent niches. And so you could have either species present, it's doing the same thing in the ecosystem, even though it's a different taxa name, its metabolic position is the same. So that was the idea we were going for. Okay. So we do a bunch of computational biology, which is not particularly interesting, but it ends up at the point that we have a set of reads that we got from the rumen, and we match those to a bunch of enzymes which code for reactions. And when we do that, we can get a picture like this. So let's pause for a minute because I know this looks horrific. It's not as bad as it looks. Each one of these little dots is a reaction. In particular, it's a reaction on this side of the network for which we have an enzyme sequence. So, or more than one enzyme sequence. So we take our reads and we say, aha, we've got a bunch of reads from each animal that map to this enzyme. And if we colored the dot green, that means more reads from the grass-fed animals map to that reaction. If we colored the red nodes red, that means more reads from the grain-fed animal map to that reaction. And if they're black, they're approximately equal. So each dot is a reaction. And we join reactions, although I know you can't see this very well with lines, when they share metabolites. Okay, so far, so that's actually the metabolic network idea that I just showed you. What we add to that is the following. First, we add the host metabolic network. So this is actually the metabolic network of the sheep. Each one of these dots is, again, a reaction that's encoded in the sheep genome. So we know that by comparative genomics effectively. And then what we do is we get a list of compounds that are potentially produced by the microbes but absorbed by the sheep. And so in this case, this is mostly the 20 amino acids plus 3 short-chain fatty acids. So those are the things that are being produced over here by the microbes and potentially at least absorbed by the host genome, the sheep metabolic network. We can then effectively sort the metabolic network into layers. So this is a layer that produces the amino acids. This is a layer that absorbs the amino acids. And then this is sort of one layer of reaction removed, 2 layers, and so on and so forth. So at least conceptually, this is not quite how it's really going to work, of course, but conceptually you can imagine the diet of the sheep coming in over here. So these are the compounds that are coming in the diet, the components of the grass. They're getting broken down and broken down and broken down by the microbes until they are broken down into compounds that the sheep can absorb. They cross this boundary, roughly speaking, and then they get processed further by the sheep to make DNA or whatever the sheep is going to do with that, either for energy or for biosynthesis or whatever the sheep is going to do with those compounds. So essentially, these are compounds that are sort of shared between, these are reactions that are close between the host and the microbes, and these are sort of very distant microbial reactions. And so then we can start to ask questions about what do we learn from this. And the first thing we learn from this is that effectively the weight of mass of the metabolic network of the microbes is different between the two diets. So if I go back here, I can sort of show you that. The animals fed grain tend to have the bulk of their reads in this part of the network. The animals fed forage, fed grass, tend to have the bulk of their reads in this part of the network. What's going on there? Well, the idea is these sort of starchy grain-based diets consist of a bunch of compounds that are pretty close to the host metabolic network. So we're sort of bringing the diet in maybe here, and it just has a little bit of a distance to travel to get to the host. On the other hand, the grass diet is cellulose, all those lignin, all those things that are actually really hard to break down. So it's using a bunch of reactions that are quite distant from the host, and so that diet has to go through more layers of the network before it actually hits the host. That's exactly what we see in the real data, so that's actually rather encouraging. So we're saying that the forage diet is sort of more complex, more distant from the host-based diet, and so we've got to sort of work over here more before we can send those compounds into the host. The concentrate diet, the grain diet, is already pretty similar to what the host can absorb on its own. So that's actually a nice result. The second result that we didn't expect is, and this is pretty hard to see and I apologize, I've basically shown the same network twice, but what I'm showing now, color coding the dots by essentially their variance. So if the dot is bright red, it means that that metabolic reaction varies a lot in read count, animal to animal. And what we saw was the 8 animals fed the grass diet had pretty similar metabolic networks. There wasn't much variation animal to animal in read count. So all those metabolic networks were rather similar. On the other hand, when we fed the animals, we again, obviously not me, fed the animals these grain-based diets, the animal-to-animal variation in the metabolic networks of the microbes was bigger. What would that mean? Well, we're still not quite sure about that. But we did, I'm going to go forward and come back to that slide for a second. We actually just said, okay, let's look at that in a different way. So we just listed we had about 3,000 reactions that we found. We turned those into a vector, we normalized it, and we calculated pairwise distances between all the pairs of animals. And so what you see is the 8 animals fed forage diets, grass diets, compared to each other, so all possible 8 times 7 over 2 pairwise comparisons. They're all quite similar, both at the taxonomic level, but also at the metabolic network level. So again, they have rather similar metabolic networks. But when we look at the animals fed grain, they're all over the map. Some of them are really, really different from each other. So these pair of animals have very, very different metabolic networks. So they're fed the same diet, but for whatever reason, they've built very different metabolic networks, microbial ecosystems in their ruminants. Why might that be? Well, we don't really have any idea. These blue dots are essentially when we compare a green dot to a red dot. So these are the difference between a forage metabolic network and a grain metabolic network. Now what you can see is there are individual pairs of grain-fed animals that are as different from each other as some grain-fed animals are from grass-fed animals. So what that's suggesting is that somehow we can build a lot of different microbial ecosystems that are very different taxonomically and very different metabolically, but maybe function about the same way. Now, there are I think at least two interpretations of this data. The obvious interpretation is this is the evolved diet of these animals. So this is what they're really trying to get to, and this is essentially what happens when you force feed an animal something it really shouldn't be eating. That's an attractive hypothesis, but I don't think that's actually what's going on, because I lied to you slightly when I described the experimental setup. The experimental setup is everybody started out on this diet, then these guys stayed on this diet, and these guys got shifted. So what I think has really happened is these ecosystems got disturbed. Some of them sort of recovered, and some of them didn't. And in particular, if you look really closely, and obviously you wouldn't have done, there are 4 little dots down in this region, which are 4 animals fed grain that actually have rather similar, actually, sorry, 4 dots, 3 animals. 3 animals that were fed grain that actually have very similar metabolic networks. Paradoxically, those are the animals that responded worst to the diet shift. So they didn't gain very much weight when they shifted to this grain-based diet. And when you go in and look at these metabolic networks, what you see is the reason they're all very similar to each other is they're all very uncomplex. Essentially, most of the diversity at the taxonomic level, most of the diversity at the metabolic level has been lost. And so the analogy I use is this is a kudzu forest. Basically, you switch the diet, the ecosystem changed, and instead of responding well and building a nice new metabolic network that efficiently processes a new diet, everything sort of went to heck. And so you ended up with these animals that just didn't respond well to that diet shift. Obviously, there's a lot of people in my department who are very interested in that, because this is not something you want to have happen to your sheep if you happen to be having it at home. One thing you might think is true, but surprisingly isn't, this is not at all driven by presence absence. So you might say, well, the difference is really just there's a bunch of reactions that only get used in the forage-fed animals. There's a bunch of reactions that only get used in the grain-fed animals. That's simply not true. What this picture is showing is anytime there's a white dot, that's a reaction that was present in both diets. Anytime there's a colored dot, that's a reaction that was only present in one of the 2 diets. But when it's colored black, that means there's sort of 1 or 2 copies of that reaction in one of the 2 diets. What this is saying, and I can show you the statistics if you're curious, is there just are very few presence-absence differences between these diets. Almost every taxa and almost every reaction that we find in one diet, we find in the other diet. And again, I think that comes down to our experimental setup, which is everybody started on the same diet in the same place in Wyoming, and so they all initially built their ecosystems from the same set of organisms. And then when we shifted the diet, we didn't kill anything off, we just changed this abundance, either increased it or decreased it. So the last thing we did is we said, well, can we maybe try and reproduce this result computationally? So over here are the pairwise distances from our real animals. This is a node pairwise distance. Don't worry about this one too much. It's another measured distance. Okay, so they're over here, whatever. We then said, well, what happens if we computationally try and simulate the shift of a forage-fed animal to a concentrate-fed animal? So we basically start out with an average forage metabolic network, and we simulate a shift. We force it onto a different diet. Well, if we sort of naively force it, we actually don't get anything that looks anything like our real data. That was kind of disappointing. But then what I said is, well, all I'm doing here is I'm just trying to create a metabolic network that's, I have essentially a goal in mind. And my goal is actually a set of compounds found in the concentrate diet. And I'm just moving reads around until I get as similar a set of compounds as I can to that goal vector. So that's actually why it doesn't, we don't replicate what we see in the real data. But then we said, okay, we can't, what I was doing with that approach is I was just taking a given reaction, I say, well, let's get rid of this reaction and add a new reaction. Let's move a read from reaction A to reaction B until I get a network that looks like the grain-fed diet. No good came of that. But when I then forced myself to take pairs of reads that were connected to the network and move them as a pair back and forth. And that corresponds to, say, the death of one microbe because it doesn't survive well in this diet, and the reproduction of the second microbe that does. Then I actually get something that looks rather like the real data, but more importantly, I get this interesting separation. Apologies, and so what this simulation framework shows to us is when you take a fixed metabolic network and you change the diet, even in the simulations, you get a group of networks that are more dissimilar to each other than when you keep the same diet. And so, avoiding all of the complications, what that's trying to say is there are multiple equivalent solutions to this ecosystem problem. If I'm trying to take one diet and change it to another diet, there are multiple different metabolic networks that all solve that problem that are quite different from each other. And so again, what this is saying is that the reason that those animals that were fed grain had very different metabolic networks is not because that's not the evolved diet. It's because probably when you change the ecosystem, there are a number of equivalent resolutions to that are equally good. There's a bunch of different potential metabolic networks that all solve that ecological problem equally well, even though they're rather different from each other. Of course, you can insert your own epsilon for equally well. It's not perfect. But it's interesting because we see this also at the single organism level. There are a lot of different equivalent metabolic networks that can make their living off the same substrates. And so this is sort of, there's more than one way to do it argument about biology. And I think that's important because that means when we study any given metagenomic environment, we should keep in mind that there's probably not one taxonomic suite or one metabolic suite that would work in that environment. There are probably a whole bunch of different ones. And so we should not expect to find the same suite of organisms over and over again, or the same suite of reactions over and over again. There may be multiple different ways to do it that all work pretty much as well as each other. Okay. So, is there anything to take home from this? Well, I guess that's up to you. But I think the key conclusion of the first part of the talk is when you're looking at a polyploid organism, you have to know when in its evolutionary history you're looking, because a new polyploid will be under the pressure of different evolutionary forces than an old polyploid. And so sometimes you see these papers that say, well, this idea about polyploidy is wrong, because when I look at my favorite neopolyploid that just was created 10,000 years ago, I don't see any of those forces at work. And what we're saying is, those forces may not be at work in a neopolyploid. There'll be another set of forces that are working in a neopolyploid that just happened, that are no longer at work in an old polyploid, like say Arabidopsis. So whenever you're looking at a polyploid organism, you're trying to understand what evolution is doing to its genome, you need to know when you're looking, because that'll tell you what forces are at work. So I think the metagenomic part of this talk was one long plea for me to know more about ecology. Because the real questions, as you can sort of see, are not really genomic questions. We can map reads, we can build networks, all that kind of thing. The real questions are, should we be talking about these as ecosystems? Are they model ecosystems? Are they useful ecosystems? And you end up with questions like, is having a lot of different kinds of metabolic reactions in an ecosystem better than just having a few? Is diversity good from an ecosystem perspective? There are reasons to think that it is, but we'd like to be able to frame that question so we can test it with these kind of data. And I don't quite know how to do that yet. But that's where we'd like to go. We'd like to be able to say, yes, this sheep is growing faster than this other sheep because it has a more diverse rumen microflora that's better at processing the food into energy than the less diverse one is, and we can prove why that is the case. We're nowhere near there, but that's where I think we want to be going. And obviously we don't have to do that in sheep, we can do that in people. Except in people we always don't want to do backwards, right? We want to eat a whole bunch and not gain very much weight. So we want to be unsheep. Okay, so where are we going in the lab? The next thing I want to do in the first part of the talk is to try and look at modularity. So people throw around this idea of modularity. That is to say, there's sort of little groups of genes in the genome that work together, and you can pull them out and put them in another genome and they work in the other genome just as well because they only weakly interact with other things. Is that true or not? Is biology modular? That's kind of a difficult question. We think we could actually look at that using the polyploidy. We'd like to essentially do everything we did in the yeast polyploidy again for brassica. Again, that's broccoli, cauliflower, cabbage, kale, Brussels sprouts, all those things. We had to rewrite every line of code because this is a genome duplication, 2 copies of the genome. This is a genome triplication, and every time my code said 2, I had to go back and replace it with 3. So that was a sort of 3 months of annoyance. But it's done, so we can do that. The last thing I just mentioned that we're wanting to do comes back to something I alluded to earlier, which is this idea of, can gene duplication reduce gene expression noise? Gene expression noise is probably not mostly a problem. It probably isn't a real issue. But the one place it might matter is in development. Because you can imagine that if you have the wrong level of a particular transcription factor at just the wrong time in development, that can throw the whole developmental pathway out of line. And so my sort of really wild idea is maybe the vertebrate 2R genome duplication stabilized vertebrate development enough that you can have bigger bodies, essentially. Because if you think about development, it's a sort of a binary process, right? You're having cell division after cell division after cell division. every time that goes wrong, a cell type becomes the wrong cell type, you have the potential for a failure. If you reduce the gene expression noise in that framework, you may be able to do more successful cell divisions without failure and sort of build bigger organisms. So maybe one of the things polyploidy does is stabilize things like development. That, I think, is a kind of a wild idea, but I think it's worth testing because we can do it with some computation without having to collect data. So maybe we can find some interesting things there. So, thank you very much.
Thank you, Gavin. Questions?
Please.
Okay. It's a very good topic. So I have a very naive question first. So what is the triplication? Does it happen at the same time?
And of course, I shouldn't answer that very clearly, but no, it's probably a duplicate. It's probably one hybridization followed by a second hybridization some number of generations later. It's pretty clear that's what happened in Brassica. So you made a hybrid, and then that hybrid then crossed back to another species. And so you actually have 3 genomes playing around. We've tried to model that. We can probably see the space between that second hybridization event in our models is a bit messy.
So I have a question about the yeast duplication.
Mm-hmm.
So among all the extant yeast genes, are they all from the whole genome duplication, or there are even Asian duplication or some more recent ones? How many, if you know?
There is no proof that there is another more ancient genome duplication in yeast before the one that we can see. I have a vague suspicion that there might be, because the histone genes are paired duplicates that are older than the genome duplication. So maybe. I think that's pretty unlikely. There's certainly been a lot of post-genome duplication, extra gene duplications. So the telomeres are always growing and shrinking, and extra genes losing genes. And then, like, the hexose transporters have duplicated a number of times since the genome duplication. So the genome is by no means fixed after the genome duplication. There are subsequent losses and gains. I don't have a number for that, but it's in the hundreds.
And for the model you proposed in the duplication and loss, that similar to the HKY model,
Mm-hmm.
that one is the data just present or absent 1 zeros there?
Yeah, it's essentially, it's more complicated than that how you analyze it, but essentially each genome we say, are there 2 copies or is there 1 copy? And then we try and phase the 1 copy or the 2 copies to be orthologous across all the genomes. I can show you how to do that, but it's not interesting, but it's intricate.
I just wondered if the independence assumption there, so for example, if your genes are at different locations on the chromosome.
So the other thing what I didn't say is the reason I use John Gordon's data is all this is grounded in gene synteny. So all this is grounded, not merely in the fact these things are blast hits of each other, but they're in the same relative order across all these genomes. So yes, there could have been a gene duplication, but the idea that there was a gene duplication that happened to land in exactly the wrong place in the genome to mislead you into thinking that that's an ancient genome duplication, that probably happened once, but it didn't happen 100 times. There's probably 1 error out of 4,000, I would say. I saw another hand that disappeared.
I have a naive experimental question. I'm sure you're familiar, there's a large body of research doing experimental evolution in yeast and in bacteria by changing the environment, and we see sort of the selection of individual variants. Mm-hmm. Can one make an allopolyploid in the lab? And if so, can we watch this model of evolution transpire?
You can make an allopolyploid in the lab. I'm not sure how much it's... And so, for instance, the strain of yeast that makes Carlsberg beer is actually an allopolyploid of Cerevisiae and Vianis. They are not particularly keen on letting us look at that because it's a trade secret. But so you can definitely do it. To my knowledge, it has not been traced. I think actually a friend of mine is thinking about doing this. It has not been traced over a large number of generations yet. You also see weird things when you do experimental evolution in the lab with yeast. You see a lot of aneuploids where you get 1 extra chromosome, not a whole suite of extra chromosomes. My admittedly naive interpretation of those data are that when you have a really constant environment, the evolutionary behavior is not the same as you get in a real environment. And so you actually see this in brewing strains of yeast, which is basically you force them into a very constant environment, and by the end of the brewing process, you'll get a bunch of aneuploid yeast. If you go put those back out in the wild and try and let them compete, they die. But in that one constant environment, the aneuploid is actually selectively beneficial. So the trick is really to set up your environment in such a way that you can mimic nature enough to actually see this play out. The people who are doing this are oddly enough doing it in brassica, and Trichopogon and that sort of thing. The plant people are doing these resynthesized polyploids. But they're particularly interested in what happens in the first few generations after polyploid. And that seems to be somewhat replicable. And it probably has to do with things like methylation and that kind of thing, where there's sort of, you bring the two genomes together and there's one that dominates and one that doesn't. So I think there's a lot of space to do that, but I don't know that anybody's actually tried to do this exact thing in the lab yet. There's no reason to do it. So I should, but whenever I'm in the same room, other people's PCRs break. So that's why I'm not a lab biologist.
I'm kind of curious, on your phylogeny, you were showing that different species were losing genes at different rates.
Yeah.
So I wonder if you can do a rate analysis and see if why you're having different rates across different species and what the pressure is for that.
It's an interesting question. The model puts that out. I could force a molecular clock, quote unquote, in the model. I didn't. So that's sort of something the model creates for free, but we don't really understand why the model creates that for free. The only thing we do know that probably is important is that this thing, which is the one that has the longest branch, is a pathogen. So it's probably sort of undergone some sort of reductive evolution. So there's something wonky about the forces that are happening in this genome relative to all the others. You also have to be a little bit careful because genome quality comes into this as well, right? So if you've miscalled, if you've missed genes, then your branches will be longer than you haven't missed genes. And these are the genomes that are less well sequenced than these. So the fact that these branches are a little longer than those could be an artifact of genome quality, or it could be that they've lost genes more rapidly. So I think there's a very interesting question there, but I don't know how to get at it.
The second question I had is subfunctionalization usually infers pretty much expression. Nobody's really looked at functionality, right? And so I'm kind of curious in what you think about the ideas of subfunctionality.
I think we've been misled. I think the expression example for subfunctionalization was an excellent illustration.
Yeah.
I think we've tended to make it the canonical pathway when it's not. I think it was intended as an illustration by Lynch and all those people.
Yeah.
For example, I think actually the opsons are an example of subfunctionalization in the sense that it's not exactly the same. Actually, Mark Rauscher's, just across the road, I think, has worked on what he calls escape from adaptive conflict. And it's sort of another side of subfunctionalization. And the idea is you've got a multifunctional gene, but it can't specialize in either of the two functions because becoming good at this function means it becomes worse at this function. You can resolve that by duplicating and specializing each copy. That's subfunctionalization, but it's actually selected subfunctionalization. Because, well, yeah, but what's interesting is the classic model requires the fixation of the duplication by drift and then the divergence. This model says the duplication becomes beneficial almost immediately because selection can now act independently on the two genes to diverge their functions. And so if you think about like the opsons, where you had the situation in the primates where you had a red allele and a green allele, and they were at sort of heterozygote balance, then you duplicate that. You fix one locus for red and one locus for green. That, I would say, is something like subfunctionalization, but it's subfunctionalization of protein function, not of expression. So I think there's a group of people who are reading the subfunctionalization literature very narrowly and saying it's always drift, it's always expression divergence. I don't think it is always either of those things. And I also think there's a danger in assuming that the fact that you see tissue divergence and assuming that was selected. It may be that that gene doesn't need to be in that tissue and just got lost or got gained. So you should not assume that because a gene is on in a particular tissue, it's needed there. It may just be on in that tissue because it's not harmful.
Yeah, that's been pretty much the argument against
Yeah, so my interpretation of this DCC model was that's duplication, degeneration, and complementarity. It was intended as a mental model of how you could have some force to fix duplicate genes other than neofunctionalization. It was never intended to claim that was what was causing all gene duplications and fixations. But some people have wanted to make it that.
Any other questions?
Thank you so much.
Thank you.